Welcome to the pod Biomanufacturing and Fermentation Technology. In this episode, we translate microbial science, process engineering, and manufacturing realities into insights that scale.
Okay, let's unpack this. We are diving into a really crucial, high-stakes phase of industrial biomanufacturing that I think for a lot of people is almost invisible. It's that moment the fermentation ends, that supposed victory lap where the vessel is just full of product.
And that victory lap is exactly where the most valuable material, the very product you spent maybe days or weeks creating, is actually at its most vulnerable. We're doing a deep dive into what's called post-fermentation biological state control, which is, well, it's truly the silent killer of your yield in these massive operations.
I love that phrase, silent killer, because you spend all this money, all this effort optimizing the cells to maximize production, and then boom, instantly the entire goal just flips. It becomes about minimizing change.
Exactly, minimizing all biological and chemical change. But when you're dealing with a massive manufacturing vessel, that flip it's never instantaneous. You can't just hit a pause button.
Right.
The research really drives this point home by highlighting the physical reality of scale. In a lab, sure, harvest is pretty much immediate. But in manufacturing, you're battling massive thermal inertia.
So like trying to cool a swimming pool versus a coffee cup.
That's the perfect analogy. And you have these long transfer delays and all that residual biological activity. It keeps going. For hours, the broth is still a highly reactive system.
So the core problem is really that if you don't instantly shut down that residual activity, it starts to eat away at the very product you just made, often before downstream processing even gets a look at it.
That's it, in a nutshell. And so our mission here is to really explore how harvest timing, these so-called arrest strategies, and the hold conditions, how they determine whether your upstream gains are preserved or, as we said, silently lost.
Okay, let's start with the engineering side of it. Why is harvest itself such a high-risk engineering problem? I mean, on the surface, it sounds like it should just be simple plumbing.
It's anything but simple. The sources clearly define this as the point where residual biology, your product chemistry, and, critically, your facility's limitations all collide. The end of active growth doesn't mean the end of biological risk. It actually triggers the most dangerous phase of vulnerability.
I have to keep adjusting my mental scale from, the lab bench.
Yeah.
When you say the system is still active, what's specifically happening inside that giant tank, even after they've turned off the feed?
Well, residual metabolic capacity is still there. The cells are burning through their internal fuel reserves, and that supports low-level activity. That means continued heat generation, which is really hard to remove, and continuous redox shifts in the broth.
And what else?
Well, on top of that, if the cells enter a stress state, they can start releasing extracellular enzymes. Or even worse, they start to lyse. Just a little, dumping all these destructive intracellular components right into your product mix.
So the product molecules themselves are facing this degradation clock that's measured in hours, not days, which means your harvest timing and your temperature control become absolutely decisive.
They're the controls that matter most. The decision to terminate a fermentation is a serious engineering control action. It's not some casual endpoint. And because these large vessels cool so slowly, and it's not even the center is hotter than the perimeter, that shift from a growth-optimized state to a preservation state is a very gradual transition that has to be actively managed.
Okay, so if an instantaneous harvest is a myth at this scale, how do engineers actually figure out the optimal moment to start that stop sequence? I mean, just relying on a single sample from a port feels incredibly risky.
It is, and that's why it demands multi-parameter logic. You need integrated indicators that tell you what the entire culture is doing, not just what's happening in a single sample tube.
So what are these integrated markers? What gives them that big picture view?
We're talking about monitoring the culture's respiration patterns, specifically the off-gas. They look at things like the carbon dioxide evolution rate, CER, and the oxygen uptake rate, or OR. These rates will plateau or inflect when the main substrate runs out, signaling that the culture has metabolically shifted away from active growth.
So reading those gas inflections is sort of like catching the culture's last gasp. What other indicators back that up?
A sudden collapse in base demand is another strong one. It signals the end of net acid generation, which is tied directly to their metabolic state. They'll also track the growth rate asymptote, which shows biomass accumulation has slowed way down. But the really critical overlay for this whole decision is what we call the product-specific stability window.
That stability window, that must be key because if you wait too long trying to squeeze out every last gram of product, you might actually lower your final recoverable amount. Is that the danger of delay?
That is precisely the danger. Here's where it gets really interesting. Industrial data consistently shows that a delayed harvest right after that metabolic plateau can just tank your recoverable activity, especially for sensitive products like therapeutic proteins or enzymes.
Wait, okay, so if I'm an engineer and my mass-based assay, the test that measures quantity, says the titer is still high, how do I even know I've already lost value?
Because those mass-based assays are blind to functional damage. You might be holding for more titer, right? But if the cells are stressed, protease activity just skyrockets during that stationary phase. These proteases are literally slicing up your product. And while the bulk mass is still there, the active fraction, the part that actually works, is decaying rapidly. The number on the screen looks great, but the functionality underneath is just silently being destroyed.
That is fascinating and genuinely alarming. Okay, let's dig into those destructive mechanisms. What exactly is powering this value erosion inside the vessel during that hold?
The primary force by far is proteolysis. The culture doesn't instantly freeze, right? It enters that post-stationary state. And any of those released or extracellular proteases become major liabilities. They just keep acting aggressively during the transfer and the hold periods.
And we know rapid cooling is a challenge, so how sensitive is that destructive activity to heat?
Highly sensitive. Enzyme activity accelerates dramatically with temperature. It follows what's called Arrhenius-type behavior. This means that if you hold the broth at, say, 30 degrees Celsius instead of your target of 10 degrees, that pretty modest temperature bump, it accelerates activity loss significantly over a typical 4-hour hold. Your product is being chemically digested in slow motion.
So the key distinction is that the little cleavage fragments are still counted in a protein assay, but they're functionally useless. Okay, what's a secondary mechanism of loss?
The second major concern is oxidative modification and redox drift. This is absolutely critical for oxidation-sensitive products. Think about proteins with sulfhydryl groups or enzymes that need specific metal-binding sites.
And where is that oxidative risk even coming from if the metabolism is winding down?
It's a combination of things. You've got some residual oxygen exposure, and that's combined with redox active species, like certain metal ions or cofactors that get released during cell stress or that mild lysis we talked about. That combination drives irreversible oxidation, and that changes the protein's shape.
This uncontrolled redox drift, as the sources call it, it sounds like a one-way street toward creating impurities.
It is. It's predictable. It increases your impurity load before clarification even starts, which in turn just crushes the yield you can capture downstream. You're effectively generating tons of process-related impurities after the fermentation is done, but before purification begins.
Wow. So you've got temperature-driven proteolysis and chemical-driven redox drift working together. Let's shift to the solutions. What levers can engineers actually pull to stop this, knowing they're constrained by the cooling rate of a giant tank?
The fundamental strategy is rapid cooling. It's the most universally effective lever because it just slows down the reaction rates for almost everything at once.
But scale kills speed here. A pilot system might cool in minutes, but a manufacturing vessel takes hours.
Precisely. And this is why the sources really stress that the limiting factor isn't your final temperature set point. It's the integral of time temperature exposure. That means your product is degrading all the way down that cooling curve. The engineer's job is to minimize the area under that curve.
Okay, that makes sense. But if proteolysis is so dominant, why not just dump in a chemical protease inhibitor? Is that just a cost issue, or does that create a new headache for downstream?
That's a great question, and it really highlights the trade-offs. Often, adding a standard inhibitor is just prohibitively expensive when you're talking about 50,000 or 100,000 liters. But more importantly, it introduces a new component that has to be removed during purification. That adds steps, it adds regulatory burden, and it can even interfere with your chromatography columns. It's a coupled problem.
So they have to rely on physical and sort of generalized chemical strategies. Besides cooling, what else is in the toolbox?
They can look at oxygen management. You can intentionally crash the dissolved oxygen level to suppress that aerobic metabolism and the heat it generates. But you have to be careful. Shifting the redox environment too quickly might actually induce an unfavorable stress response, leading to more cell lysis and more protease release.
A real catch-22. And the other main lever mentioned was pH shifts.
Yes. Adjusting the pH can be very effective at reducing the activity of certain proteases, or it can physically stabilize the product molecule itself. But again, there's a big trade-off. Shifting the pH can risk precipitation, which creates viscosity problems, and then you need complex neutralization steps later on. It just complicates the whole downstream process.
So what does this all mean? The governing principle for the engineer is that the best strategy isn't what works perfectly in the lab. It's the one that manages that risk within the facility's existing and often slow constraints.
Exactly. If you know that cooling is going to take 6 hours, then maybe a stabilizing pH adjustment early on, even though it adds complexity later, is the lesser of two evils compared to losing 30% of your functional yield.
And sometimes, those holds are just unavoidable because of scheduling conflicts between upstream and downstream. So time itself becomes this critical, managed process variable.
That's just the reality of large-scale manufacturing. And doing these intermediate holds, your product integrity isn't just governed by time and temperature, it's also governed by those residual chemical factors and, critically, by bioburden.
Bioburden amplification. So if you get a little contamination during the transfer, that nutrient-rich broth is just the perfect place for it to grow quietly.
It's a silent, rapid amplifier. A very low level of contamination can support explosive microbial growth during that hold, and those contaminant cells release their own potent proteases, drastically increasing the degradation risk before you can even see that anything is wrong.
So during a hold, your standard growth phase metrics are basically useless. What new diagnostics do they have to rely on to track this degradation?
They need diagnostics that are aimed at function, not just quantity. The sources really emphasize how essential routine product activity assays are. You cannot rely on a total protein assay. You have to measure the functional biological activity because that's the only thing that tells you the loss is happening without a change in mass.
Function over mass. Got it. What else?
Off-gas and pH drift monitoring are still vital. If you suddenly see an unexpected pH shift or renewed CO2 generation during a cold hold, that is a massive red flag. It means unexpected metabolism, or even worse, contamination. And for oxidation-sensitive products, you have to be tracking dissolved oxygen and redox potential continuously. It's non-negotiable.
So what does this all boil down to for the listener who's looking at their bottom line?
It means that if your facility reports hitting a fantastic end-of-fermentation titer, but the final purified yield is always variable or consistently low, the most likely culprit is loss during those intermediate holds. The main yield loss happened pre-downstream processing. It was a failure of biological preservation, not necessarily failure of purification efficiency.
That is a huge reframing. We should stop blaming the capture steps for losses that were actually baked in during those hours between stopping the feed and starting clarification. The signature is always that discrepancy between mass and function.
Yes. The common scenarios of loss, they always center on thermal exposure. That uncontrolled time-temperature integral and residual metabolism altering conditions before you can start physical separation. This isn't just about scheduling, it's about recognizing this phase as its own unique, highly critical unit operation.
And speaking of critical operations, if the industry is still struggling with this, what's the main gap preventing better control?
We still lack robust predictive models that can integrate all these moving parts. We need models that can realistically simulate protease kinetics, oxygen exposure, and the actual temperature history, given the limitations of industrial cooling systems. This would allow engineers to predict exactly how much yield they'll lose based on a decision to hold for 4 hours at 12 degrees versus, say, 6 hours at 8 degrees.
So the data is there, but the ability to model that complex, hours-long reaction trajectory in real time is really the current frontier.
It is the next major step in recovering hidden value from these processes.
So what does this all mean? The focus can't just be on getting the microbe to produce more. The focus has to shift to engineered preservation. The moment you stop feeding, you start the clock on yield erosion, and that clock is ticking a lot faster than you think at scale.
Productivity means absolutely nothing if you can't functionally capture it.
Treating harvest and those intermediate holds not as simple logistical nuisances, but as engineered unit operations with defined acceptance criteria and response logic. That is the essential step to ensuring those upstream productivity gains actually translate into final recoverable yield. The next time you look at a biomanufacturing facility, remember that the most intense process engineering often happens not during the growth phase, but during the cool down. What you make in the reactor is just potential. What you recover determines your success.
You've been listening to Biomanufacturing and Fermentation Technology with Prasad Ernala. Thank you for joining me. Until next time, Keep engineering biology into reliable manufacturing.